So I forgot to bring my camera stabilizer, so this is going to be a little bit rough. But because this week, or this month, what everybody asked for in the poll on my Patreon is a breakdown of kind of the things that I'm working on here at the Odin. And as you can tell, I'm here in the cell room again. So I'll go over some of the stuff that we're working on in here first. So obviously, I'm still working on the stuff with the fish. You know, we've got zebrafish and goldfish, and the fish project in general is partly learning and partly preparation. So we're going to be working on trying to create like a microinjection kit that people can get, or like a microinjection class that people can take, so that we can teach microinjection as a skill, but we need to make things as easy and reliable as possible before we do that. So, you know, working with zebrafish because that's sort of the standard, but we're also trying to work with the goldfish because they're much more available and they're way easier to care for. Goldfish are like super cheap, super easy to get, super easy to care for. You know, they're robust, they're tough, they're just harder to breed, and that's sort of a problem. So we're trying to figure out how to breed them easily. And so, you know, we're working on that. With the zebrafish, the main thing is just making sure that we can like do everything reliable. They're just much harder to care for. They're much more fragile fish. And that will move into doing microinjection with mouse embryos when we're done with that. And that will move into doing some of my cloning stuff for my personal projects. I've covered a lot of my personal projects, but not so many of my work projects. But that'll go into microinjection and genetic modification of mouse embryos, and then reimplantation of those embryos, which will lead to eventually working on trying to grow those embryos without using a mouse. So that's going to be like an artificial womb sort of thing, but that's probably well into next year, maybe a couple of years from now before we even get there. So we want to make sure that all the foundation work and all the steps between here and there are really, really solid. Let's see. This also ties into my cloning stuff because obviously the microinjector can be used to make genetic modifications, and I can make genetic modifications other ways also, but the microinjection rig is going to be really useful in moving the nucleus from the somatic cells, which is like the cells that are just like grown off the dog, into the actual embryo. And the other cool thing is that we're doing in here is stem cell work. So this one's pretty new. So we're going to be trying to we also have, I guess before that. So the other thing we're doing in here is we're working on cultured meat, right? So we have like a fish cell line, and that one's nice because fish as a tissue culture grows at room temperature. They don't need like an incubator or anything, so that's easier and more accessible for people because you can just grow it on your kitchen counter. And the other thing is we're looking at chicken, which we already have an established chicken cell line. It's just a matter of like how do you scale and what else can you do with it and those sorts of things. But the stem cell stuff is sort of tangential to that. The stem cell work is we're basically trying to make sort of human cell lines. So eventually you'll be able to get like a stem cell class, right? But or like a stem cell kit, and it'll come with some stem cells. And so those stem cells will be sourced from like me or Josiah or both. So you could buy like David cells or Josiah cells and experiment with them. And it'd probably be something along the lines of, you know, like you could buy some of my cells and they'll come with a kit that'll have like growth factors and things like that. And so you can grow them starting as stem cells and transform them into other types of cells. So you can transform them into, you know, nerve cells or muscle cells or like cardiomyocytes, right? So heart muscle cells, which are really cool because like if you do it right, they'll spontaneously beat. You'll just see them under the flask in the microscope going boom, 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 which is pretty cool. Or like nerve cells that can like grow and communicate and maybe even grow like organoids, which have like little brain structures. And so stuff like that that we're looking at moving into and you know, offering to people so that people can like get some experience with growing stem cells and things like that from, you know, a source that's not like wrapped up in IP, because you could just buy my cells and do whatever you want with them for the most part. Just don't do weird stuff. But one of the things we'll need is actually not in the cell room because I have to do some work on it. It's actually way over here. So we're headed to like the main lab area, right? We're also gearing up a lot for the holidays because there's going to be a big rush. We're going to have a big sale, so there's a big rush of orders that always come in the holiday season. So we're getting ready for that. But let's see. This guy. Oh, so I can angle the camera. This is an electroporator, which is going to be necessary for the stem cell stuff, because these stem cells will be derived from our blood, and so you collect blood cells, separate out the cells that are actually nucleated and those that aren't, and then genetically modify those cells, and you got to modify them with some high efficiency. So that's where this guy comes in. Electroporators are great for modifying difficult to modify cells or modifying them with a really, really high efficiency, but it's missing some components, so I've got to like build a cuvette. So that's like high voltage stuff, so that's going to be fun. Let's see, what else have we got? Yeah, so I'm working on that. You can see other little things I'm doing, like repairing some of this old equipment, trying to get everything like the home lab kits and stuff out, because some people can get them by Christmas. But you know, we're just gearing up for a big rush of sort of holiday orders, so like everybody else. But let's see. So I've got to work on the electroporator before we can do the stem cell stuff. And I've got to get some new media and things like that. We've also got some other peripheral projects going, like in the mycology department here. So this is mycology stuff. So this is like the whole mycology area. That's our new flow hood. And this, so we've got like bioluminescent mushrooms and stuff, but it's still like mostly mycelium. It's crazy how the distortion on this camera works. That thing's huge, but it makes it look tiny. I guess if I get next to it, you can see it's actually very big. But yeah, so you know, we've got some different things growing. We're building you know, a lot of different stuff, getting like better medias, and really trying to break down what it takes to grow these things, not just like grow them, but to really grow them, because mycology's full of growing stuff on like wood and barley and, you know, things like that aren't really defined media. And so if you want to create something that's really, really reliable, that you could put in the hands of anybody who's not necessarily a mycologist and it'll really do well, then having access to that, you know, having done the work to make sure that all of those protocols are really, really reliable, and that what you're growing them in is really ideal and optimized and really made in a way that is repeatable and reliable and is identical time and time again, a really defined media. So that sort of thing. And we're also working on like trying to find ways to reduce contamination, because contamination is obviously a big problem when you're growing anything for a long time, because contaminating organisms have a lot of opportunity. And so trying to find a media that will select for the bioluminescent fungus and select away from, you know, contaminating fungi and bacteria and stuff like that. So, you know, it's a bunch of stuff like that. I'm also helping with Kessler. So Lyra is doing the mushroom stuff for the most part, and I'm helping. And I'm also helping Kessler. And Kessler is working on our biomaterials stuff. And so that's another thing the electroporator will be good for, because the species of bacteria isn't, you know, or probably isn't specifically easy to transform with normal protocols, but electroporator is really good for those sorts of cells. So, you know, we can be able to make genetically modified cells that make different biomaterials and interesting biomaterials, and, you know, biomaterials that are reactive and responsive to stimulus. So, you know, they're like green and fluorescent, or they're, you know, fluorescent when you shine a light on them, or they're, you know, keratinized when you add whatever, and then you can make like harder biomaterials and biomaterials with different properties and all those sorts of stuff. So it's really interesting because the ability to like grow a hard substance is really interesting to me, because then you can like grow shapes and things. So there's a lot of stuff going on in that space, and that's mostly Kessler, but also I'm helping. I get involved in everybody's projects. Let's see. So also got about the only thing I'm not involved in really is like the algae stuff, and we're gonna have some plant stuff coming up soon. I say soon, it may take a little while. Plant stuff is always slow. But that's sort of the roots of what I'm doing. Most of the stuff that I'm doing is animal-based stuff. You probably could have guessed that. So it's like, you know, embryo injection, cell growth and development, stem cells, that sort of stuff, tissue culture. And I'm also working on the human cell class. So the human cell class up until this point has been, you know, you culture human cells, HEK cells, and you genetically modify them to give them resistance. But I'm tweaking the kit to make it better. So hopefully removing some of the things to simplify, and then replacing the DNA, the plasmid, so that instead of just giving them drug resistance, you give them something that allows them to change color. And I don't have an example with me right now, but what I'll do is I'll like try to edit in a picture somewhere of the new results of the kit. So that should be coming out in January. There's gonna be like a live class. Josiah will be teaching it. So what will happen is right now there's like a live version of the kit that you can get. And so in like January, we'll ship out a bunch of those, and then you can like do it step by step with Josiah live, and ask questions and that sort of thing. And that will replace the current videos with the new videos filmed during that live class. And that'll like transition everything over to the new kit, which will be sort of the human cell kit version 2, which is a lot of what I've been working on also. The thing I'm most excited about right this minute is the stem cell stuff, because it's super new, something I've never messed with. The stem cell stuff is really interesting because I really am excited about the opportunity to culture my own cells and then transform them. So I have briefly cultured my cells before, but that required a biopsy, 6 of them actually. And I don't want to do any more biopsies because like taking a chunk out is just not fun and I don't like it. So in order to avoid that, that's what this new protocol is about, because it only uses a blood draw and I'm comfortable with that. So, you know, if I can do that, then I can also do that with other organisms. So at the same time, I'm going to be drawing blood from Osiris and attempting to culture stem cells from him, or at least culture cells. They don't necessarily have to be stem cells, but that would be interesting. But as long as I can get a good solid culture of cells and preserve them, then I can, you know, have access to his genetic material whenever I need it. And also, you know, I can create, hopefully, that'll feed into the cloning project later on down the line. But it should be pretty interesting. It's really fascinating, all the stuff here, because it's a really sort of high creativity environment, like everything's very experimental. So while we do a certain amount of things like with the standard protocols, we also investigate the protocols a lot. That's what we spend a lot of time doing, which works good for me, because that's what all my personal projects have always been, is like just trying to optimize and make more accessible protocols that are otherwise really complicated and difficult. And so it's really like trying to find the bottom, trying to find the minimum necessary protocol to do things successfully. And that requires a lot of creativity. It requires a lot of exploring the space. It requires a lot of like trying things that probably aren't going to work. Because the trouble with a lot of these protocols is that they are, a lot of these protocols are already optimized, right? But the criteria by which they're optimized are efficiency and things like this. And accessibility is usually not a big factor. And doing it as cheaply and easily and quickly and accessibly as possible with the least learning curve and difficulty and most reliability isn't usually the criteria, right? So trying to optimize for like something that's accessible and digestible to the general public is a really interesting idea, and also I think really necessary. And it's also something that I think, gives you an opportunity to kind of explore a lot of the standard practices, and it gives you an interesting perspective on the nature of those practices, because you kind of get a sense that, oh, this is the right way to do it, but for a lot of things, it's not necessarily the right way, it's just the way that it tends to be done. And the way that things tend to be done often is great, but many times it's unnecessarily complex or unnecessarily expensive or unnecessarily inaccessible for some other reason, like you need an expensive piece of equipment that you don't actually need. And so those sorts of things, those sorts of spaces are really interesting to explore, because, if you think of it as like a local minimum, right? That's where a lot of these protocols end up falling into, right? Because they are optimized for these other things. The protocol itself then becomes the norm. And then and anything you do that changes it slightly generally reduces its effectiveness, right? So, if you think of it like a rubber sheet, right? Or like a gravity well, right? The space has been explored to find the bottom, to find whatever the optimal is. And of course, optimal means different things in different circumstances. And so generally speaking, if you deviate, you change your incubation time, or you change your media, or you change your whatever, sometimes it has no effect. Sometimes it makes things better, but generally speaking, it makes things worse. And that's because these things have already been optimized. The trouble is, you can be in a local minimum, right? You can be in a little bump, if you think of it as like sand dunes, or you think of it like rolling hills, right? You drop a ball somewhere at random, and it rolls to the bottom of whatever hill it's at and finds some minimum. And that would be like, and if you were the ball, and you sort of explored the space to find the bottom, you roll in this direction, you start going uphill. It's like, oh no, that's not where the bottom is. And you go this direction, you start going uphill again. You kind of go every direction, and no matter where you go, you're going uphill. So everything you do is kind of worse. And so if you're only exploring in these little increments around this minimum, you seem to be making things worse. Every change you make makes it worse. So you convince yourself that that's the right way, it's the optimal, it's the perfect way. But what you might find is that at your minimum, this little bowl that you're in, if you were to go all the way over, there's an even lower minimum, right? So you're in a local minimum, but if you can explore all the way into this crazy direction, you find that there's an even more optimal way. And a lot of times that requires doing things that are wrong. So it's like, okay, well, if you increase the incubation time from an hour to an hour and a half, it's less effective. If you go 2 hours, it's less effective. But if you go 17 hours, holy shit, it's amazing. And that sort of thing, it usually doesn't work, right? But when it does, it can make things really, really interesting, and it requires doing things that you know are wrong or you think are wrong. It requires doing things that are crazy, that are just really out there and creative. So there's a lot of trying crazy things. It's like, okay, what if we grow them like this? What if we do that? What if we substitute this for that? Or what if we just don't add that? What happens? What if we try to grow them without this and without that, and throw away all the things that everyone says are needed until you get to a point where you find out what really is needed and what can be substituted. And sometimes you can do without one thing if you add something else. And so there's huge space to explore around all of this stuff. And so it's a huge thing to try and, adjust. It's like, think about all the wheels you can adjust, all the different ways you can make changes, because there'll be like 50 knobs, and each of them has as many settings as you can dream of. And so trying to perfect that sort of thing, it's almost impossible to find the perfect conditions for any of this, because the options are too great. The space to explore is too vast, which means you're almost guaranteed to be in a local minimum and not in a universal minimum. You're not in like the perfect protocol, you're just in a pretty good protocol, and so anything you change makes it worse until you change enough things to make it great again, and maybe find an even better, more optimal protocol. But that sort of thing is really hard. It takes a lot of creativity, it takes a lot of work, it takes a lot of like willingness to fail, and that's the biggest thing, is you have to fail a lot. You have to do a lot of things that are definitely wrong, and usually they are wrong. But sometimes stuff works for crazy reasons, and it just feels like magic. And I think that's probably the most fun part, is doing things that feel like magic, but it gives you a really beautiful, deep understanding of the stuff that you're working with, and it gives you a lot of power over the things that you're doing, because you really understand every little element. You're not just running the protocol because that's the protocol, and you don't know why the protocol is what it is. You know why all the things are what they are. You know what they do, you know why they do it, you know what changes are gonna lead to what, and you've thoroughly explored the space, at least as much as is reasonable. No one can fully explore any of these, really. And that's the kind of thing that I think is the most interesting about it all, is the idea that it's unexplorable. It's just too great, it's too vast. It just cannot be fully explored. And that sort of thing means that you have such endless possibilities to play with, and that there's not many things that are not worth trying, right? Some things are not worth trying, and you can kind of think of it from first principles and have a sense of what those are. But sometimes you have no idea. And sometimes you just do things that work for other things, or, and that's one of the big reasons I like to be involved in everybody else's projects, because, the more I learn from one person's project or another person's project, the more protocols I have in my brain, the more possibilities I know about, the more things that I can cross-apply from, one piece of biology to another. And so that's a lot of what I do. That's a lot of what I work on, is just making sure that, first of all, that this place has all the things that it needs, and then that we can create new things continuously to give people access to things that they don't have access to, in ways that by solving the problems that are standing in their way, basically. And that's the cool part. So, okay, I think that's everything. My arm is super tired from holding up my phone.